job of setting them up with the TVG gainer or whatever he tweaked.
Oh, yeah, totally.
Yeah, they weren't working that
think?
well before.
Also, Kate, you're setting us up really good. I like it.
Yes, she is. Perfect.
So did you see that rock indication there, front row?
Say it again.
So if we could get a 20 centimeter cubic rock, it looks like
Okay.
Emil just circled that one. Did you see it?
Yeah, I did, and that looks, yeah, I'm with you.
So
Dan?
park with your left.
Left, okay.
Yeah, use the magnum or left or whatever hits on the left. So give me some room with the manipulator, otherwise it'll be too tight. Crab in there a little bit.
You ever think of putting something like a ski pole on Hercules so you can just like poke it out and grab, just a toe hold type of thing?
Something like that.
I just imagine Hercules skiing for some reason.
What's that?
I just imagine Hercules skiing. Steve was like, attach a ski pole.
So, sorry, which was the victim here?
Right to the left of the lasers.
Okay.
Gosh, I love that little drawing thing. It's so great.
Nice.
Yeah, sometimes that goes out over the live feeds and sometimes it doesn't. So
Oh, really? Okay.
Oh.
yeah, it seems like today we're getting some reports that from viewers that they're not seeing it. So I'm gonna have to
Oh, rats.
do some investigation, but we got a telestrator here and the stylus is in the hands of the scientists. So
It's a loose tooth.
It's a loose
You got
tooth.
it.
you
can
got
get it.
this. More beans.
I believe it might be bigger than it appears.
Yeah, it might be bigger than it looks.
What does it mean that the jaws are actually able to scratch and scour?
They're mostly able to scratch and scour the manganese crust, which is, because it's a precipitate, is usually really actually soft.
That's
okay.
Big one.
That's
So that
big.
might be too
That's a little big.
that might be too big to be
Left
the
it out
neighbor of the
around.
one that's already in there. Yeah.
Oh, but we know how much Dan likes picking up and throwing down rocks. So.
So yeah, I guess that guy's too big to get into the box with the
In F,
other one.
What-
Yeah.
I don't know.
Okay.
Yeah. We can see another angular one behind this
We're
plume.
just at a stop now.
Yeah.
Can't see a lot.
Yeah.
I don't know about you,
So
I can't see
bouncy.
anything.
It hurts my eyes. Oh.
So we got a special message for the team Tectonic Turtles from a 4-year-old viewer
Oh.
who is watching the feed as part of bedtime.
All right.
This is, you say rocks tell the best stories, and in this
Yes.
case, we've got some bedtime stories here. So
yeah.
thanks so much for writing in. It is our pleasure to be coming to you live and to be sharing this ocean exploration with you.
I think we've got
May
an
your
honorary.
sleep be as deep as the seafloor.
Oh,
Yeah.
Emil, you're a poet and you didn't even know it.
I think he
Any
does
other,
know it. He stuck it right in there.
Any other likely victims here?
Yeah, let's see. What's angular? Can we reach up here anywhere?
Yeah. should
This
be able to.
guy?
We're on a cliff. Pokey there. Push Gabby off the cliff. I saw that one move. When it might be embedded a little. That one's definitely loose.
Does he look cubic when you pull him out?
It's angular.
It's angular.
It's a little flat.
It's a little flat.
Yeah,
Yeah, don't
Yeah,
like that one.
No, he's too thin. He probably altered through.
that one fell flat.
Fell
What was the
flat,
other
but
one?
bump.
Was this guy here, was it?
Yeah, try him. Oh.
Oh,
Oh.
That might
Oh.
be hooked on.
that appears to be solid.
Okay.
We have
How
time
about right here?
Oh,
Steve, are you looking at the fish?
Yeah.
oh, great view on Argus.
Here, I'll get you a...
Fishy friend is
Oh, yeah.
between Herc and Argus.
It's looking
Let's
pretty
see.
good.
That
Steve,
one
I'm
looks-
driving that camera now.
Yeah.
Okay, cool.
Little small.
Little
Oh, it's
small.
kind of rounded.
Kind of rounded. Okay. Sorry to be a pain in the buttocks.
I can pick up rocks
How about-
all day.
Okay, how about that one? Right.
Don't throw me in the briar patch. Whatever you do.
Is he angular and chunky? I can't tell from this view. That's a problem. I can't tell. I can tell you pick them up, right? How thick they are.
Yeah, me neither.
Oh, man.
Yeah,
okay.
no, that one's
It's definitely a 2D world.
Yeah. Okay, let's see. Do.
Pen
Yep.
up just a hair, Gabby.
You
Say
see
again?
one?
Or tilt up just a hair.
Yep. Okay.
If you see one, Dan, that you think is cubic,
Oh, I don't know. You want a small one though, right? Not a big one?
yeah, you want it. We want like a 20 centimeter guy.
That one's not 20 centimeters.
What if I were to pick up and put down like a couple feet to
How
the
about some-
left?
Pen left a little?
Okay.
Yeah, maybe a little reposition.
Oh yeah, that guy might be good.
Oh,
Anybody have eyes on that fish? Oh, there it is.
It's there.
Yeah, he's there.
Oh, it's so close.
I'm waiting for it to swim in
okay. You
right
can
in
move.
front of her.
Bridge now.
There
Is
we
that
go.
Dan?
You can move.
I can move. Okay.
Can
Can you go full
wondering why
wide on
you're
the
moving
Argus?
towards
Steve?
the
After that.
northwest?
Full wide.
Was that the guy we abandoned because he was too big right at the porch? Yeah.
Okay.
Yeah.
I think so.
Our eyes were bigger
Great,
than
I
our
didn't catch what
sampling
you just
box.
did. So,
I didn't do anything, but he just drifted
Oh.
about 20 meters, and I just called up and asked him why.
okay. And what happened?
Copy. Yeah, you just drifted probably about 20 meters, so just calling up to see if there was something up with your thrusters or not.
Are we going to the left here? Which
Yeah,
way are we
Let's do
going?
it. Let's
Yeah.
do it. We got time.
We don't have too much time because we might have to chase the ship a little bit.
We drifted the
Oh,
right way.
so-
I've been watching the Argus sonar. So.
But that one by the claw right now.
Which one's by the claw right now? This guy?
Put it down there. Gabby.
Yeah.
Yeah.
I can do this one on the fly.
Okay.
There are strong winds, so they're adjusting their heading, but I don't know why we drifted.
Is that guy too big?
It's falling
Oh, it
apart.
just broke off. Oh, no, it was
Oh,
just
it's-
sitting on top of it?
I think it's massive, right?
Pretty big.
I don't know.
It's about 20 centimeters, I think.
Where is our lasers? Oh, I see it.
That fish is still hanging out.
Oh,
I
You're up to 20 knots now, and it's risen probably about 5 knots during 4 knots, yeah.
let's see. That guy looks pretty good.
Like, why
Yeah.
are we
What do you guys think of that guy?
That looks
but
like a
We
big
like
size for the big
Yeah.
bucks. It's not too flat. Is it?
No, not too flat. It's a little thicker. I think that's a good chunk.
That'll go in the small drawer.
Yeah.
Or with
Then
that
the
other
big-
one.
Yeah, it's going to go in the big
F.
drawer.
so going with F.
Yeah.
Box
Yeah.
Yeah, come off the cliff there and then dead stick. And take your
So it's a nav. You say the wind is coming up.
Or disable that.
Yeah, the
starboard
winds come
thrust.
up about, oh wow, actually 5-6 knots since we went in the water. Yeah, and the ship was having a little bit of trouble holding position, so we drifted
Okay,
north
open just enough
while
for
we
the big
were, I
bucks.
was assuming we were standing still.
That fish is still there.
Could just be
He's
a gust.
coming into the light if we...
Yeah, we don't want to let the sponge out there.
Forecast was calling for gusts to 20, so maybe it's temporary.
Open a little more.
Yeah.
I did tear that gasket off of there. I think I got spares. Okay. There's one for the red book.
there's a nice rock sample. And I think that fish is still above Hercules on the Argus
Did you,
view.
Yep.
Did you turn your thruster back on? I'll do the camera if you want.
I wonder if
on the
he's
box.
eating stuff associated with our sediment plume.
Or with our lights.
Oh, yeah.
Although things at this depth are probably more likely to run away from the lights than run
Oh, he's
towards the
getting
light.
closer now.
It's hard to know. Well, height he's at though? Like, is he just outside the lights or is he above? Seems like he's above.
He's definitely high above
Just above,
Hercules.
yeah.
Clear water on your left.
He definitely hasn't left. He's been hanging out.
I've never seen one of those things actually feed.
You have not?
No, never. The rat tails, they just hang around. Must eat like once every 10 years or something.
Should we keep moving?
Yes, I think so. Can we go along the same transect?
Probably same thing, 195, 30 meters.
Yeah, I like this. We got a little more room there. We're about 30 meters away from the cliff. It's a little more comfortable.
Yep. Yeah. Bridge, nav. Can we please move 30 meters, bearing 195? Thank you.
That coral there that looks like a precious coral is occupying 2 orientations in relationship to the current. Yeah.
So is that one?
I wonder if that means that the current isn't as strong here, that it doesn't force them into that directional orientation as much.
What is that red thing above the pink coral?
That's another coral, I think.
Soft coral?
Yeah.
Is it?
Apparently.
Oh, yep, it looks like a soft coral.
Yeah, the soft corals are so neat.
And there's our
little shrimpy,
primitive lobster.
Yeah,
this one looks more more narrow than the
more shrimp leg, maybe.
Maybe.
We're having some issues with our stream for our viewers at home. Thanks for letting us know about that by submitting in our website. We know there are some issues and we're sorry about that. We're going to do our best to get that sorted with our folks in Rhode Island.
I just wanted to see the soft corals there.
No, this seems to be a channel 1 issue. Yeah.
Okay, go on.
It's intermittent, but both our viewers and scientists ashore are reporting it. I submitted a video alert.
I'm getting a little far away here, just getting carried away.
So we're talking about these corals in relation to the currents. They are filter feeding organisms, and so if you picture something like a net, they're trying to cast as wide a net as possible perpendicular to the current so that they can catch small particles of food. And in previous dives, we've seen them all lining up in the exact same orientation, which gives us a good idea of the currents, but these past couple of collections, it seems to not be as straightforward.
Yeah, I'll drop down to 10 meters
good.
and give you a little more.
Yeah.
What's the coral on the top screen? Top of the screen?
Coral at the top of the screen?
Yeah.
Okay.
Just a name would be helpful. that the bottle brush?
Not sure about that. Maybe so.
I'm not familiar with a
I'm
bottle
going to
brush.
go with some type of chrysogorgid.
Can I get a zoom video?
Ooh.
Nice.
Thank you.
Okay, go on.
Just gonna zoom in on Argus there a bit.
Okay, thanks.
I got focus failure there, do I, Steve? I don't know how long that'll last. It makes it hard to keep Herc framed up.
I'd like to get a zoom on one of the thick fans here.
Oh, yeah, okay. Cool.
It's a nice little biological community here.
I know.
Everyone is
little
different.
Collection.
We can bump the ship one more time or hold station for watch.
Let's hold station.
Roger.
Okay, Steve, can I get a zoom?
We have time for a bump. It's 20
Okay.
minutes, 20 till.
Zoom in?
Yeah, I don't think I'm going to be able to push in much further without shading the lights.
Nav.
You can always put the ROV 45 and use one of your
Can we move
one of your
bearing
eyebrows.
195?
I typically use the
30 meters.
Magnum side eyebrow,
Thank you.
so DSC is further away.
Is the one next to it a spiral one?
Yeah, I think
Yes.
so.
Come here wide.
Thanks for the zoom.
Yeah, thank you.
That'll give you the starboard side lights on it too, Gabby. We won't shade so much if you're
Okay.
not perpendicular.
Yeah. I see what you're saying, Dan.
Just beware the DSC sticks out there a bit on the
Mhm.
starboard side. So.
Yeah, I feel like I don't have as much dexterity to land on those light bars. I find that very difficult.
I do all the time. Padded. That's what they're there for.
We're getting some nice diversity here.
Yeah.
But usually on the cliff like this, it'll touch the bottom left.
Okay.
The Magnum also we can. It's a good handhold.
So a viewer wants to know how we decide which corals to just look at and go ooh-ah, and which ones we sample for further study.
Get a lot of help with some coral experts ashore and on board.
They tell us the ones that they don't currently have samples of, because in order to identify a new species, it's not just by the morphology that you see, the big picture morphology, but they actually look at things under the microscope to identify new species. So in order to do that, they need to have something that they can examine and characterize on many different levels. So looking at it from its morphology down to its microscopic characteristics is one of the reasons why we will sample something. So if we identify something that looks like something we've never seen before, or that is not part of the catalog of organisms that we currently know of, that would be a good a good candidate for potentially sampling if we see more than one.
Some of these corals are easy to identify just by looking at, and some not so much. incredible zooms we're seeing from video on those corals, showing the little polyps, help identify a lot of these.
We did an expedition years ago in the Discovery Corridor off of Halifax, and we had an array of 40 boxes on the front of the vehicle for coral samples.
Whoa.
Wow.
We were allowed no more than 10 centimeters per sample, and that was a whole DNA study they did. Filled all those boxes.
So here's a nice input from Scott France. Most of the deep sea corals we know of were described in the late 19th and early 20th century, way before we had video. So
Interesting.
characteristics used to ID the species don't necessarily match up with what we see in the videos. So we're learning and need samples to make the connection from the microscope to the video.
I've got a question. I might have missed the discussion about it, but do we know why this is named Don Quixote Seamount?
I do not have background on that one. Maybe somebody thought exploration of it would be an impossible dream.
And
maybe
here,
you thought it looked like a windmill?
Here we are tilting at a windmill.
Oh, sponge.
Oh, a little
wee
spongy.
sponge.
Little wee sponge. A little sponge that could.
The GEBCO database claims that this seamount is named after an early ship visiting Hawaii in 1840.
1840.
Nice. Googling.
What happened?
Steve requests that the next time we see a reasonably high density or diversity that we get a Niskin, please.
Okay.
We're tugging some way. Why are we tugging? I'll come down 5.
So we do have limits on the number of samples that we are allowed to take, and it depends on if it's something that we know of or if it's something that the scientists aren't sure of and think it might be a new species. Our permit to operate this expedition in Papahanaumokuakea Marine Monument is very specific about not just the biological samples that we collect, but also the rock samples. There are limits to the quantity that we can collect on each dive. So we just try to collect the best of the best and get the samples that will give us the most information and the most new information.
Yeah.
Yeah, for each
come down to
ROV dive, we can collect 15 rock samples, 5 biological specimens per species.
That'll get us a little
but then there are
targets.
some further restrictions based on the abundance of them.
Yeah, we're usually more limited by the space that we have in the bio boxes and the sampling boxes on the vehicle. We try to fill them up, but they are limited in size and number, and oftentimes, especially with the rock samples that we're collecting for Beth for microbial analysis, we don't want to cross-contaminate with other samples, so they kind of have to live alone. So the permit does have limits, but usually our most important limit is the size and quantity of our sample containers.
And also we're respectful of the uniqueness and the sacred area that we're in. And so we try to sample for the best that we can do, meaning the best example of a given organism or the rock that gives us the best chance to test our hypothesis so that we can do it sparingly.
Yeah, and mindfully. I think that's what if you've been listening for a while, you've heard us conduct these protocols. I think that's kind of the intent there, is that we're mindful about our collection.
Can we move 5 meters, bearing 315?
So as we get closer to the end of our shift, we got another great question from a young viewer out there. Will Nautilus still be operating in a couple of decades when I'm old enough?
Pulling on Argus there pretty hard.
Couple of decades?
I think we all hope so.
Yeah.
Oh, maybe a bigger, better.
Yeah.
Maybe a bigger, better Nautilus.
Nautilus 2.
That's right.
Nautilus 2.
Right. I'm
Yeah,
30
there's
meters
other
away from the cliff
ships of
now,
exploration as well. If you're interested in this, there are opportunities in many roles, marine biology and
I'm
many
30
other
meters
roles.
away from the cliff, so
And there will also be rocks coming
that's
back from Mars at
pulling
that
the heading
time
around,
that
so
you will
it's
be
making
able
you
to
struggle.
study for
Just
signs
that
of
5
life
meters
that might have
could,
existed on Mars.
make your life a little easier.
And you might not have to wait a couple of decades. We do have college internships for college students in various
I
roles.
would have done 10, but it's close to shift change.
Data logging, and we have mapping interns and ROV interns on board. Those roles are publicized on nautiluslive.org. You can apply and join us. So you may not have to wait as long as you think to become a member of the Corps of Exploration. Thank you so much for your question and for your interest in this work. You're why we do what we do.
That Mars comment reminds me of a good question I have for everybody in the van, but I think I'll save it till our next watch.
Okay. We're eagerly awaiting.
Speaking of the next watch, over the next few minutes, you will be hearing various members of this watch, the Tectonic Turtle Watch, Turtle Power. You'll be hearing us go off SPL so that we can brief our replacements. You'll see that also on Sat Feed 3 as they're coming in. We let them know where we are and what we've been doing and what we've been seeing, and then we'll hand off to them. This is your 12 to 4 shift.
Yeah, it's tracking well.
Yeah, thanks so much for watching. And this dive is expected to continue for about 8 hours. And see some more amazing things.
The ship moved heading a little bit, and the sonar dynes, I think it's just lined up better because if it's about there, right? And now we're not, I think before it was on the wrong side of the cliff, like the west side of the cliff. Yeah. So we like couldn't reach them. Yeah. Yeah. Or somebody was just messing with it in the gym, who knows?
Really nice.
Mm-hmm.
Keep your questions coming for the next watch at nautiluslive.org. We are available to take your questions and comments. We especially love hearing from students out there. Thank you so much for your questions. We really do appreciate it.
Just going to do a quick pre-shift gauge strafe. All right with that? Gauges are good.
Yeah. So if you're interested in learning more about how to get involved with Nautilus and the careers that could lead you here, you can go to nautiluslive.org and click on team. You can see how each member of our Corps of Exploration got here. And I think the Cliff Notes version of that is there is no one path. We have many different specialties aboard ship. Some are scientists, some are not. And so check that out on our website and find the path that looks the most interesting to you. Lots of ways to get involved and lots of ways to make this your work.
I think the one common factor in those different paths would be good teamwork.
Absolutely. And also I think curiosity.
Yep.
You know, you can persist through lots of challenges if you're really, really interested in knowing answers.
Like,
making discoveries.
Maybe a few animals there just to the left a bit.
Yeah. Passion. Yeah. Thanks for that zoom.
Looks like it just
a
goes
nice
around
sponge.
the corner there. Maybe up a bit. Some.
Thanks so much, everyone. I'm going to be signing off here. I have been your Science Communication Fellow for this dive, and all of our other roles you can see us swapping out on Sat Feed 3. Thank you so much, and our replacements will be back with you on SPL in just a minute. Enjoy the dive.
Bumping your camera once.
Thanks.
Bumping your camera for you. Thanks, everyone. It's Dan signing off.
Good afternoon, everybody who's watching. You just were treated to commentary by the rock stars, and now you're going to get commentary from Team Sponge.
Hui Huakai. That's who we are. Hui Huakai. Team Sponge in the house.
Okay, do you want, while we're stopped here, do you want to do our ceremony, or do you want us to move along and do that in a minute?
I think we are, is everybody just about ready?
Give me 1 second here.
Sure.
I'm almost ready. Yes, we are.
Well, good afternoon, Team Sponge. How are our pilots doing? Our navigator, can you guys hear me? Our video engineer.
Yep, we can hear you.
Yoo-hoo. Our scientist, our lead scientist, Chris.
I can hear you. I just couldn't talk to you until I pushed my button to turn my mic on.
And
don't make me come back there.
Our data logger.
Yeah, doing good.
Great. And our wonderful Andy.
Yeah, I'm here.
Excellent.
Aloha.
So, good afternoon, viewers around the world. We are here with Team Sponge, starting our watch. And before we begin, we need to set our minds, bodies, and souls to our mission. So we are going to go radio silent to conduct a private ceremonial protocol to acknowledge and honor the biocultural resources that are present and that we are interacting with and impacting. So we will be back on SPL shortly.
All right, we are back.
All right, thank you. Okay, shall I get moving here?
Alrighty.
So I was told in the transition we were following a feature. It was kind of tapering out, but they recommended we go up the slope just a little bit. Does that sound like a good thing to do from your perspective there, Science?
Yes, I think so. So I'm taking a look at your HYPACK screen right now to see where we are. Yeah, that would be fine.
Okay.
Yeah, some very nice deep features here. Our team Tectonic just before us, they were finding some very unique habitats and some nice sponges and corals, and collected quite a few rocks for their sampling.
Yeah, this site looks like it has possibilities of providing for something for everybody.
Mm-hmm.
Had some wonderful rocks. I was looking at those when we first landed, and so yeah, I was sure Andrea and the geologists were quite excited because they're easy to pick up and they were beautiful pieces of pillow lava.
Nice.
It is really an amazing little outcropping here.
So Trevor, do you want to keep the ship on the kind of on the contour it's above, and you just move up, or do you want to step closer? It is pretty close to the wall as far as Argus.
It's pretty close to the wall, yeah. Let's we can do 10 meters closer for now and see how it goes.
Okay, so you want...
Or do one of these actions, 45 elated, whatever.
Yeah, sure, that sounds good. So how about like a 210?
Sounds great.
215? Okay. Make that 215. Bridge nav. Can we get a 10 meter step bearing 215? Thank you.
So again, wonderful rock is a very subjective term. We have two different geologically focused missions on this cruise. One is to date these seamounts, and they are looking for angular rocks that aren't too weathered. And then we have Dr. Beth Orcutt and the work that she's doing with the microbes on the rocks, and she wants weathered kind of squishy rocks. So two very different purposes, two very different collection methods for the different science that they want to conduct.
So from what I've seen so far, I think Andrea is going to win this rock fight. There hasn't been too many squishy ones. These things look really good for dating, but I haven't seen all that much for Beth's. But you never know. As we get a little closer toward the top, we might see something.
Yes.
That'll work for her.
And a squishy rock is one encrusted with ferromanganese nodules, these cobalt ferromanganese crusts that we're looking at, which is a deep sea process where these metals precipitate out of the water and attach to the rock. And what Dr. Orcutt's looking at is what role microbes play in that process. Do they enhance the process? Do they speed it up? Do they slow it down? Are they getting energy from these things? We don't know about these microbes as we head into a world that is looking to harvest some of these minerals, because the areas that they are being harvested from on land are diminishing or in unstable areas for harvest. So they're potentially looking at harvesting them from the deep sea. But we really like to know what's down there and what these organisms are doing before we start this deep sea mining process. It's already slowly moving ahead. They don't have the equipment yet to be able to do it, but there are leases along the Clarion and Clipperton zone in kind of the central eastern Pacific, and those are being leased by different countries around the world, and they are moving ahead with developing this equipment. So we really want to try to understand these processes, but of course that won't happen here in Papahanaumokuakea Marine National Monument, because we are 580,000 square miles of protected ocean here in the central North Pacific. And we're also considering, NOAA is considering a sanctuary, a National Marine Sanctuary designation for this area. So if you want to provide your comments on that, now is the time to do it. You can go to our papahanaumokuakea.gov website, where you can participate in that, provide comments straight through the end of January, or attend one of our meetings, the virtual meetings we're going to be hosting in December. We'll host 4 public virtual meetings, and you could provide comments on what you'd like to see for that sanctuary designation, or provide any type of comments about this special and unique place that we've been working in.
What do we have?
Yes.
What do we have? We have a Team Sponge mascot showing up.
Sponge
This
mascot.
little wee guy?
It is an indicator.
Can you zoom in on the wee guy, please?
I can.
Hui hua kai. Yes. Imua. We move forward.
So what I think I'm seeing, and I I might go out on a stalk here.
There is no stalk to go out on, Chris.
Yeah, I have to see what the side of this thing looks like, but this is possibly, it could be one sponge that settled on another sponge. Is the one on
really?
But I'm not positive because I can't see what's actually going on since we're looking at it head on.
I'll try and get over to the side here.
Okay, thank you. Oh, it's two separate sponges.
Oh,
That's
cool.
what's going on. You tricky guys.
And no stalk
yeah.
on either. So Chris, tell us about some of the indicative features that tell you how to be able to identify these sponges.
Well, these days I use a technique that's referred to as Gestalt.
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 All right.
And what that means is I've seen so many of these sponges in other animals over the years that sometimes when you see an animal, you just know it. You're almost unconsciously looking at a diagnostic feature, but it's not really going right into your head. You just know that's one of those. Those two sponges were both the same species, and they are in the family Pheronematidee. They're glass sponges.
Okay.
And they're at least for now, they're in a genus that we call Poliopogon.
Poliopogon.
Yeah, say that 3 times.
Yeah,
Actually, I like that word.
Poliopogon, but can't do it.
And this one coming up too is yet another member in that family, and it is either an older version or it's a different one. And these are beautiful, massive sponges, and they are fascinating to look at under a microscope because they have a very distinctive microscopic part or a spicule that a lot of sponges don't have, and it's called an amphidisc. And it looks like 2 umbrellas that are in opposite directions. The umbrella part's in opposite directions, and they're connected by a single handle right there. So it's I love seeing those things, and I don't know why. It's one of my favorite spicules when we put something under the scope.
Amplidisc, you called it?
Excuse me?
You called it an amplidisc?
It's called an amphidisc.
And what does that do?
It does. Who knows what it does? I mean, who knows what a lot of these spicules do? But the fact of the matter is, there are not that many sponges that have got that type of spicule. So it's a very diagnostic spicule for a certain group of sponges there. And so if I'm absolutely not certain of the sponge and I look at it under a microscope, I see one of those, I go, aha, okay, I can disregard 2/3 of the sponges in this glass sponge group.
Can you explain what a spicule is, please?
Yeah, a spicule is, there's a couple, in corals, the same kind of thing are referred to as sclerites. In sponges, they're called spicules. They're very tiny, but sometimes they can be quite big, microscopic parts that provide structure to the animals. And spicules are made of silica, and in this particular group, the glass sponges, and that's why they get their name glass sponges. And most of the time, you have to use a microscope to see a lot of them. Sometimes they can be very, very big and very long. And actually, when you look at those ones that anchor onto the substrate with this kind of tuft, those tufts are the ends of very long modified spicules.
Okay. I think that's going to be lame.
So.
Oh, just to the top of the ledge. Okay. So just want it like 10 meters east?
Yeah. The sclerites in corals are not made of silica. They're made of a very different type of material. Usually I think it's some type of calcium carbonate. But anyway, but they're serving kind of a similar process. They're providing some structure to the individual polyps in corals. And they're also very diagnostic for the taxonomy in corals, just like spicules are very diagnostic to the taxonomy in sponges.
Sounds good.
So often you can't exactly determine the species until you actually have a sample and determine the spicules, the type of structure.
Yeah, that's right. And why it's absolutely critical when you're going to take a specimen is that you get very, very good imagery, not just of the whole colony, but like, for example, corals, you zoom in on branching points and the live polyps themselves. And then when they actually examine it microscopically, and with corals, they're actually doing more genetics these days. But then you know, okay, aha, that's what this thing is. Now I've studied it in the lab, and now I'm going to look for other ones in the field that look just like it. That's the only way we're ever going to learn how to identify these things in the field without, I mean, we can't collect everything just to figure out what it is. So that's the game.
That's the game.
At what depth are we right now, Chris?
We are at 2,375 meters down.
So the other thing you were saying yesterday about these sponges that set me off for trying to identify them is that some of them, the stalk comes in and it attaches to the bottom of a concave surface. In some cases, it actually goes inside and it's a convex surface on the underside where it attaches. You have other diagnostic features such as, well, what I would like to term as a dimple on the top, whereas others are smooth.
Zoom in, please.
Some have stalks, some don't have stalks. And we're all here looking at the NOAA Okeanos Explorer Animal Guide. So you could follow along and be your own armchair explorer and identify some of these organisms for yourself as you're going along. That's available free on the website, the Okeanos Animal Guide. Check it out and try to identify some of these critters. If you do, before you hear us say anything, type it into the chat and let us know. We'd love to see what you have to say about these animals.
And that animal, by the way, that we were just looking at very closely is in the family Chrysogorgia. Oh, we got a fish. We got a fish. I'm going to shut up and we're going to look at the fish, and then I'll come back to the coral.
You can zoom on the fish.
Oh, can we get some pictures of that fish? How big is that?
See if I can paint him with the lasers here. Whoop.
We need a close-up of the side of the fish first, if that's possible, and then, of course, the head. But the side will be very helpful if you can get that.
So if you're looking in the guide, you can check out eel-like fishes. That's probably where it's going to fall in. Maybe one of the ophideiform type of fishes.
That's correct. It is an ophidiid. Oh, bonk.
Ophidiid.
But that's smarted. But anyway, yes, it's a cusk eel is the common name for this fish. And there's a couple things that you need to look for. And Dr. Ken Sulak, who's been joining, and he may be even on right now, is a specialist in fishes, and he's been very helpful. I'm going to go out on a limb. I did not see the feature on the lateral line pores along the side of the fish that I was looking for. And I look at other things such as the size of the eye, the size of the nostril and its position relative to the eye, length of the filaments, the fin filaments coming off of the bottom of it. It's got kind of a big nostril. It's right in front of the eye. If you see that little hole. Yikes. Okay.
That's all I got that way.
I think I've struck out twice today with Ken. I think he's got it, but this could be either something that's called basogygus, and that's what I think I'll stick with. And then Ken, if he's watching, hopefully he'll either confirm or
I think the
say
watch
I'm
before
wrong.
us saw one of those as well.
What's that?
I think the watch before us saw one of those as well.
Popular fish in this area. And we did have a comment also from the science portal about our discussion earlier about the the mineral exploitation in the Clarion and Clipperton zone. And Amy Gartman says there are no leases that countries can exploit in the Clarion Clipperton zone or anywhere else in the area. There are contracts that allow exploration, but there is nowhere in the area with approved exploitation contracts. Those regulations are still being written. So thank you, Amy. That's really helpful information.
Yeah, and what the stage of that growing or new industry is that the International Seabed Authority is leasing I think it's 5 mile or 5 kilometer by 5 kilometer lease blocks there and elsewhere, and then different companies in different countries can then request a certain number of those blocks. And if that's approved, then then they have to go in there and do an exploratory phase. And if whatever it is they need to determine from the exploratory phase, if that moves forward, then they can go ahead and exploit their particular lease blocks.
That's all within countries' economic exclusive zone?
No, it's all in international waters, and that's why it's actually occurring, or it's actually being overseen by the International Seabed Authority. That's where the name comes from. And here's an interesting fact. Where is the ISA located? Well, they struggled to find a neutral location for this organization, and they settled on Jamaica.
That seems like a good place.
Yep.
Can we have some zooms on these, please?
Yep.
What do we got here? Well, we got one Chrysogorgia, two Chrysogorgia.
Yeah, I wonder if we can get a side view of this particular primnoid. This is a primnoid, and I'm trying to go a little further than that.
I do have more zoom if we need it.
Yeah, go ahead.
I wish they would close up a few of their polyps. Come on, guys.
Work with me here.
Yeah, work with me here, because the way they close up their polyps sometimes can be very helpful, but, well, in the back there, they've got a few. It's the direction in which they close up their polyps, whether it's down along the stalk or up towards the end of the branches. And I see on the main stem in the back, it looks like some at least are pointing a bit down.
Sorry, I'm a little bouncy.
That's okay.
So I think we're looking at the genus Narella. And let's see, we've got Asako and Scott France who are now typing in our chat. And I think we're going to get a good ID in a moment. And Asako is asking, is this upward? And I'm not sure, Asako, whether it is or not.
It's a big one.
Scott thinks, I think the regular dichotomous branching supports Narella, so Scott thinks it's also Narella.
There's an anemone too.
This is, I think, Actinostolid anemone. It's in the family Actinostolidae.
We got to see one of those eating yesterday. Yeah. That was quite fun.
So Scott France is a professor at the University of Louisiana Lafayette, and Sako Matsumoto is a professor at the Chiba Institute a researcher in the Chiba Institute in Japan. And both of them are very well-known coral experts.
Chris, what area of Don Quixote are we exploring?
We're exploring the northwest sort of corner of Don Quixote.
Thank you.
So who named it Don Quixote? Do you know that?
I thought I heard some comments on the last watch. I was in the lab actually doing a little maintenance on one of the critters down there.